PhD in biological engineering at Massachusetts Institute of Technology, or MIT. And then he moved to Harvard School of Public Health to do his postdoc with Sarah Fortune. And has recently been appointed Assistant Professorship at MIT, where he'll be studying Flynn Lab next year, which is quite exciting for him, taking on a whole new avenue of his own research. His main interests lie in the intersection between infectious disease and systems biology. So he really wants to work on developing and understanding the dynamic interactions that occur between the host and the pathogen in TB infection, and then using novel, high-throughput, single-cell tools and computational biology to really dissect the heterogeneity that exists within infections. Bryan will be around for most of the week, so if you want to meet him, just let me know and we'll set up a time. He's also going to be giving a workshop tomorrow between 10 and 12 on RNAseq methodology for single cells, looking at some of the advantages for different systems and how we look at the methodology and can develop methodology. So if you're also interested, let me know and we'll get you a place. Otherwise, over to you.
Great. Thank you, Anna, and thank you all for being here today. It's so exciting to actually go to an audience that actually knows more maybe about TB than I do. So as Anna introduced, I'm finishing a postdoc in the Fortune Lab, and I'm really looking forward to sharing with you a story that brought us to single-cell RNA sequencing. We kind of started with this model system where we encountered tremendous amounts of heterogeneity and wanted to develop a systematic way of dissecting that complexity. So fortunately, today I don't actually have to do any introduction about TB, but I'll share with you my vantage point when I think about TB infection, which is I really like to think about the innate immune system in TB. And really, I'd like to think about the macrophages as a central hub during infection. So they're one of the primary cells that's infected. They integrate both signals from the adaptive immune system, and both have a paracrine and autocrine response that helps shape the kind of the dynamic immune context that really influence the course of infection. So one of the papers that really inspired how I think about TB and think about the complexity of infection was this work from David Russell in 2012. And so what he wanted to really understand was the dynamics of both bacterial killing and bacterial growth. And so what he did is he basically did the following. And so by classical colony-forming units, he took murine, bone marrow-derived macrophages. And what I want you to take away from this is that in the first 2 days of infection, by colony-forming units, he sees a log decrease in the total number of viable bacteria. But intriguingly, when he looks by electron microscopy and enumerates the total number of bacilli per cell, what he sees is there's a simultaneous increase. And so we see this something similar where we always see this kind of monotonic but very slow increase in primary human monocyte-derived macrophages. And so when we saw this data, what we actually hypothesized is that perhaps we have two different subpopulations of macrophages. Some that actually can permit bacterial survival and some that are successful at killing the bacteria. So that's the hypothesis that I started my postdoc off with, that we have different subpopulations of macrophages, those that permit bacterial survival and those that restrict bacteria. So in order to really test this hypothesis, what we did is we built a transcriptional reporter of bacterial viability. So let me just walk you through how this works. So we're taking classical H37RV that we're engineering to carry a plasmid that has an inducible tetracycline-driven GFP and then a constitutive red fluorescent protein. So what this allows us to do is look at infected macrophages and identify infected bacteria by looking for a red signal, and then at particular time points we can add tetracycline and ask which bacteria have the ability to induce GFP, and we use that as a proxy of viability. So we made this strain, we made multiple different colors, but the one I'll talk to you about today is this red fluorescent protein strain. And so as you can see here, only in the presence of the plasmid do we see the expression of our red fluorescent protein. And as you would predict, only in the presence of the plasmid plus tetracycline do we see the expression of GFP. So our system works, and our system's quite homogeneous in broth. And so what we wanted to test was that there was this single-cell variability in bacterial fate. So let me just introduce to you our culture system. So we're using primary human monocyte-derived macrophages, either from a CD14 positive isolation that we then mature for somewhere between 7 or even 21 days. And then we infected a low multiplicity of infection of about MOI1. And we allow infection to progress for about 4 days prior to the addition of tetracycline. And then we use high-throughput quantitative microscopy to assess bacterial viability at the single-cell level. So how we do that is we take our macrophage, we search for the presence of our M-cherry signal, and so we do that, and then we find our nuclei, and then within that border, we're actually going to say this is actually an infected cell, and then we create a second border, and then within that border, we look for the expression of GFP. So for each individual cell, what this allows us to do is have a metric of bacterial viability. So on a single cell level, we can measure GFP versus RFP ratios, and that's our metric of bacterial viability. So again, our hypothesis is that there are macrophages that can and macrophages that cannot restrict bacteria. When we actually look at primary monocyte-derived macrophages infected with the strain, what you see is tremendous variability at the single cell level between some macrophages that appear right here, this one that's got no inducible GFP, so we're calling this a restricted macrophage, and over here we see another macrophage that's got all the bacteria inside of it, and all of them seem to be expressing GFP. So we don't only look at this by microscopy, we can then quantify for every individual cell the distribution of bacterial viability at a single cell level. And what I want you to take away from this is there's this tremendous heterogeneity, and we think that this is really an effective killing because as you look over time, only over the course of time do we see this reduction in the ability to induce. So this is really exciting. We have this hypothesis, and we now see that there's variability in the bacterial survival, but really what we wanted to understand then was mechanistically, what are the determinants that really describe whether a macrophage is going to be capable of permitting bacterial survival or killing it. This raises a few questions that I'll kind of go into in the rest of the talk, is really how is MTB being killed by the macrophage? What are the pathways required? How is restriction achieved in the absence of an adaptive response? This is important. We're not adding interferon gamma. We're not adding any exogenous cytokines in order to improve bacterial control. And really importantly, what are the features that distinguish a macrophage that can kill and one that cannot? So when I think about this, when I think about we've got a dead bacterium inside of a cell, I like to posit three different scenarios. So and the way that I think about this is, what reference frame am I sitting in? If I'm sitting in the reference frame of an immunologist, I might think about the strong macrophage model, which is like a well-tuned innate immune response that's really poised for killing the bacterium. I actually happen to sit in a bacterial genetics lab, and so we oftentimes think about what are the bacterial factors that drive either survival or the lack thereof, and that might be your weak bacteria model. Or scenario 3, which is equally plausible, which is the tug-of-war model, where you have this epic battle between the host and the pathogen, and only one survives. So I think all scenarios are equally plausible, and I actually think it's probably a mix. It's really scenario 3, but the work I'm going to tell you about today is really focused on understanding the host determinants of bacterial variability. And so in order to dissect this, we wanted to use the tools that we had available to us, which in our case, in our BSL-3, we have the ability to sort macrophages, and we've got our live dead reporter. So we wanted to leverage these tools in order to really understand functionally what might distinguish these two subpopulations of cells. So in order to do that, we just basically modified our protocol slightly. So instead of quantifying by microscopy on day 5, we're actually just going to sort based off of bacterial state. So we're going to sort out 3 populations. We're going to sort out the macrophages that continue to have live bacteria, those that have dead, and those that are uninfected. And so this provides us with a lot of data that we then analyze by RNA sequencing. And instead of focusing explicitly on genes, what we were interested in focusing on were pathways, to really try to get a synthetic understanding of what are the pathways that are converging to really drive these differential responses in the cells. So we employed 2 different techniques in order to do that. So we calculate what we call an overlap p-value. So we're going to take different published gene sets. So these are gene sets that are well curated, and they say, in response to this, let's say interferon gamma, these are the genes that are upregulated. We have our differentially expressed genes from comparing macrophages with live TB versus ZTB, and then we can calculate a p-value for how statistically significant is that overlap. And then simultaneously, we can calculate what we call an activation score. So from these curated gene sets, we actually have some estimate for what we expect to be up or down regulated, and then we can look for concordance with our actual measured data. So for each individual pathway that's in this database, we can then ask for the overlap p-value and activation score. What I'm going to show you next is a summary of that data. So this is a bubble plot. And so in the x-axis, we're just organized by pathway alphabetically. On the y-axis, you're looking at activation score. And so the ones that I have colored in achieve a particular p-value. And what I want you to take away from this is there's a number of different pathways that are being differentially activated between permissive and restrictive macrophages. And the first question you want to ask is, are we recapitulating any of the known biological pathways that seem to be known in response to TB infection? In fact, when we look over here and we look for something canonical like TNF, which you know is produced by infected macrophages, we see that it's over here, but we don't really see that it's discriminating very strongly between permissive and restrictive macrophages. So it really caught my attention and might be catching yours, is what's this pathway over here that seems to be really highly activated in restrictive macrophages? And when we saw what it was, we got really excited, because this turns out to be the pathway for GM-CSF, granulocyte monocyte colony stimulating factor. And that got us really excited because there's a whole body of literature to already implicate that GM-CSF was perhaps quite important in regulating the immune response to TB. And so just briefly, there is a GM-CSF knockout mouse that has been challenged with TB and is hypersusceptible. But the caveat that I want to draw your attention to with a GM-CSF knockout mouse, it suffers from a lung defect called pulmonary alveolar proteinosis. So it's really hard in this mouse to really decouple the kind of the lung physiology from the actual bacterial effect. In mice as well, NKT cell production of GM-CSF has been implicated as important for bacterial control. And lastly, the most important piece of data that we have in humans is that the production of GM-CSF autoantibodies is associated with increased susceptibility to intracellular infections. So armed with that data, we were really encouraged, but this is a transcriptional analysis, and we wanted to really understand whether there was a functional relevance for GM-CSF in our system. So when we started this work, there was actually no evidence that GM-CSF was actually produced by infected macrophages, so the first thing we wanted to do was do an ELISA. In fact, when we look at infected cells, there's a small but significant production of GM-CSF by infected cells. And so our hypothesis is, from our transcriptional analysis, that GM-CSF is associated with increased protection or increased bacterial killing. So what we then wanted to test was whether this hypothesis was true and whether if we could add exogenous GM-CSF and improve bacterial control. And it turns out that that hypothesis is true. So here we're just looking by classical colony forming units. We see about a twofold reduction in bacterial burden when we add GM-CSF. And what's cool, and I think you'll all appreciate, is that classically when we want to get a piece of data like this, it takes about 3 weeks to get our colonies. But what was really surprising and really alleviated some of how we got through this work was this is entirely corroborated by our transcriptional reporter. So whereas this might take us 21 days, this takes us 5. And then we have this natural production of GM-CSF, so we wanted to ask whether if we neutralize GM-CSF in the absence of any exogenous addition, could we titrate bacterial viability? And the answer is yes. So we add a GM-CSF neutralizing antibody, and as you can see here, the isotype, we see this, we see a slight increase in bacterial survival when we add our isotype antibody. So seeming to suggest that GM-CSF is really important for really skewing and really driving these immune responses in the setting of TB. So now we have this input-output relationship where we add GM-CSF to our infected cells, we see a reduction in bacterial burden, but as an engineer, I really like to unpack the complexity of these systems. So, this reduction in bacterial burden really represents the integration of a number of different host effector pathways, signaling pathways that really ultimately drive bacterial control. So in the next part of the talk, I'm going to really try to explain to you how we try to kind of mechanistically unpack what this reduction in bacterial burden really represented. So I actually did my PhD in a phospho signaling network, and so every time I show this slide, my grad school advisor gets really upset because he's like, didn't I teach you better? But this is my poor man signaling network where it's been shown in literature that GM-CSF actually activates a number of downstream signaling pathways, the JAK-STAT pathway, the NF-κB pathway, among others. And we all know that there are a number of different effector mechanisms that are implicated in the innate response to TB. So given that we were working in primary macrophages, what we decided to do was use a small molecule approach to really dissect which of these pathways perhaps contributing to control in the setting of GM-CSF. So the question we're asking here is, how are these pathways integrating to drive control? So we just slightly modified our experimental pipeline, where prior to the addition of GM-CSF, we're going to add any of these small molecule drugs, and we're going to keep these drugs plus GM-CSF present for the entirety of the experiment. So I'm only going to, we tried a number of different drugs. A lot of these drugs killed the cells, so we can't really draw a conclusion there. But for the drugs that did not cause significant macrophage death, we were really surprised when we looked at the results. And so here, what I'm showing you is an example of that drug analysis. And so there are actually surprisingly a number of drugs that actually improved bacterial control. And actually some of these have been shown. For example, AKT has been shown. If you inhibit AKT, you get slightly improved bacterial control. But really, of all the drugs that we ended up testing, only this drug, which is globlenkamide, which is an inflammasome inhibitor, actually seemed to diminish control in the setting of GM-CSF. So as we all probably appreciate, humans can be quite noisy. So we then tested across a number of different donors. And actually, we've now tested maybe up to 12. And we see the effect is quite consistent. So using globlenkamide in the setting of GM-CSF seems to increase bacterial survival. And really importantly, globlenkamide is obviously a small molecule, so we use structurally similar drugs that don't inhibit the inflammasome, but are actually quite structurally similar to globlenkamide, and we don't see this effect. So we really think that this is inflammasome specific. So just as a review or introduction to the inflammasome, so the inflammasome is kind of this innate response pathway that responds to both pathogen-associated molecular products and damage-associated molecular products. What we then have is this kind of conformation here, which in response to these molecular products, we undergo a conformational change. This allows these inflammasomal oligomers to come together and form these intact complexes, which bring the protein ASC and some of the caspases in close proximity with one another. These procaspases can then become active through cleavage. These caspases can then act on pro-IL-1 beta and IL-18 for eventual secretion. And it's also been shown that that inflammasome activation can lead to a highly inflammatory form of cell death called pyroptosis. So again, the small molecule analysis suggests that the inflammasome is important. When we started this work, there was actually no evidence that the inflammasome was activated in the setting of GM-CSF. So the first thing we wanted to test was the hypothesis that actually GM-CSF is potentiating the inflammasome. So there are a number of different assays that you can use to interrogate inflammasome activation. So we tried to do all of them. So the first one we did is we looked at the formation of ASC puncta. So as these, as you can imagine, what's going to happen is that ASC is kind of floating around, kind of diffuse across the cytosol, and then as you have inflammasome activation, you're going to see the formation of puncta. So by microscopy here, in the absence of GM-CSF, you actually see very few puncta form. So you can see these red spots here. The green are bacteria, and then blue are nuclei. But really surprisingly, when we add GM-CSF, we see an incredible increase in the amount of ASC puncta formed in the cell. And below here, I'm just showing you a quantification of this. So then, okay, we get at least to this part of the inflammasome, we believe, but there are other pathways that we think that are really incredibly important downstream of inflammasome activation, namely the secretion of IL-1 beta and the induction of pyroptosis. So again, we then wanted to test by ELISA whether in the presence of GM-CSF, we saw increased in IL-1 beta secretion. And in fact, that is what we find. So across multiple donors, we find that GM-CSF potentiates IL-1 beta secretion. But then we got a little stuck because we then wanted to query pyroptosis by looking at LDH release, and surprisingly saw no difference. And so the prevailing model for inflammasome activation thus far has always been that it's an all or nothing response. That you get your ASC puncta, you get IL-1 beta and IL-18, and then you have the induction of pyroptosis. But actually, surprisingly, we found really no difference. But we were really encouraged by work by Jonathan Kagan, where similarly, they were actually beginning to see inflammasome activation in the absence of pyroptosis. So what I want you to take away from this is here, when they use LPS plus these oxidized phospholipids, they do get the secretion of IL-1 beta, but they don't get pyroptosis. Whereas if they use LPS plus transfected LPS, they do get LDH release. So what we think is that there is some way of decoupling pyroptosis from IL-1 beta secretion. However, I will say the mechanism remains incomplete. Okay, is this fully satisfying? We want to ask ourselves, are there other effector pathways that are potentially implicated and activated in response to GM-CSF addition? And so we looked at a number of other pathways. We looked at antimicrobial peptides, saw nothing. We tried to look for reactive oxygen and nitrogen species, saw nothing. The only thing where the only other pathway we saw an induction therein was the colocalization of TB with acidic compartments by lysotracker. So we at least think that two of the effector mechanisms that appear to be induced by GM-CSF are both the induction of the inflammasome and induction of autophagy vis-a-vis lysotracker staining. So in short, what I hope to have shown you thus far is that we started with this hypothesis that there's variable control by macrophages of the bacterium by using this kind of unbiased, systematic way of really dissecting the complexity of these different subpopulations of macrophages. We're able to identify GM-CSF as a critical regulator of the immune response in macrophages to TB. And then more broadly, by combining this with molecular biology approaches and kind of systematically trying to interrogate each of these effector pathways, we're able to see that GM-CSF potentiates the inflammasome and induces phagolysosomal fusion. But maybe the thing that remains unsatisfying for me.
Just a quick question. When you matured your monocytes, did you use GM-CSF in the culture?
We use 10% human serum.
And is there GM-CSF in the serum?
We do think that even as soon as you attach, as soon as the cells become adherent, they start to produce their own GM-CSF.
So you think it's autocrine?
I do think it's autocrine, but we haven't done all the experiments to really dissect.
You don't add it exogenously?
Correct. Yeah.
Did you add anything else?
No, we didn't add anything else. It's just adherence and human serum. So one of the things that really we still didn't have a good answer for is what governs the variability. So if you think about how we're doing this experiment, we are taking our monocytes, we're putting them in the same culture flask, the exact same culture conditions, yet we're arriving at this incredible heterogeneity in the immune response, then kind of in an endpoint assay, in the ability to resolve or not resolve infection. So the question I really wanted to ask is, what really is the underlying mechanism that governs cell-to-cell variation in bacterial killing? So I guess answer these questions, but we have these CD14 monocyte-derived macrophages. We're maturing them in human serum. They're all in homogeneous culture conditions. When we look by flow, and we've used a number of different markers, they look fairly homogeneous, but functionally, we see this incredible heterogeneity with regards to our infection assay. So what this allowed me to hypothesize was perhaps what we actually have is something a little bit more complex that's not being captured by our canonical flow markers. And so what this allowed me to hypothesize is perhaps what we have is a more complicated functional spectrum of MTB-infected macrophages. And so the way that I set up this framing is if we just think about it from the perspective of the ability to control the bacteria. So let's imagine that every macrophage has some probability that it's going to restrict the bacterium. So that's what I'm showing you here on the y-axis. And then there's some function of macrophage state that really underlies and tunes these macrophages in their immune response to TB. And from our functional data where we're using the live dead reporter, what we observe is that there is actually this spectrum. So we have some cells that are over here that in the absence of any immunomodulatory strategy, they're going to kill the bacterium. And then we have some where we've tried a number of different strategies. We can't get to complete control, and I think we all know this, that if we add interferon gamma, we add GM-CSF, we always have some residual population of bacteria that seems to survive. So what we really want to understand and really functionally dissect is maybe there's more complexity beyond these surface markers. Maybe there's more complexity that we really need to be able to address in order to understand really how to move any set of macrophages to a more restrictive state. So in order to do that, we embarked on a really crazy, when I started my postdoc, I would tell people that this was going to be my project, and like, do you have a backup plan? And I was like, I don't really, and so my background is actually, I trained as a mechanical engineer as an undergraduate, and then I did my PhD in bioengineering. And when I came to the Fortune Lab, I'd actually never touched a macrophage. So I kind of went in willy-nilly, and I was like, this sounds like a great problem. Let's see if we can do it. And the short answer, as I'm kind of spoiling it, we did. But it was like a labor of love, and this was a huge collaboration across multiple labs in order to make what I'm just going to describe to you next possibility. So in order to really dissect the complexity of what's going on in these infected macrophages, the first thing we did is we actually came up with a live cell sorting pipeline to actually keep sorted infected macrophages in culture, be able to sort them at roughly a low MOI. So that's what I'm showing you here. So we use a GFP expressing H37RV strain. Here you can see this is in the absence of infection. When we infect, we see two populations. We can then sort those cells out, and they're not just infected with tens of bacteria. We can actually, in fact, sort out bacteria that are macrophages that are infected with a single bacterium. And so now that we have the ability to isolate these infected cells at low MOI, what we really wanted to specifically ask the question is there variability beyond what we can see by flow? And so in order to do this, we had this great collaboration between the Shalick and Love labs and our lab at Harvard, where we were able to build a pipeline for single-cell RNA sequencing that's compatible with a BSL-3 environment. So let me just walk you through how this works. So the first half of this story is now, I guess, published, but this method is what we call seqwell. So it relies on basically making individual microwell experiments that operate kind of in geographic isolation with one another. And there's no real equipment beyond the standard pipette and the array that's necessary. A lot of the single-cell RNA sequencing strategies that exist right now require large pieces of kind of infrastructure and equipment investments. These just require the ability to have a pipette and some beads. So let me walk you through how this works. So the first thing we have is this DNA barcoded bead. So this is a DNA barcoded bead. It's just a polystyrene bead to which we have attached a cell barcode, a unique molecular identifier, and a DT25. So the DT25 allows us to specifically enrich for eukaryotic mRNAs. This UMI, or unique molecular identifier, allows us to label our individual molecules up front. Because this is still a low input method, there are a few rounds of PCR that are required. And by using this unique molecular identifier, what we can do is this allows us, when we sequence and demultiplex, we can actually count input templates instead of amplicons. So we really get a digital quantification of how many transcripts per cell. And then this cell barcode is really important because this allows us to achieve scale. So by immediately barcoding each individual cell's RNA with a cell barcode, this allows us to basically multiplex up front instead of having to use this particular Illumina adapter on a per cell basis. So this was a huge advancement because in the past, you could maybe do a 96 well plate, and that would be a cost of about $10 per cell, if not more. What this allows us to get down to is a cost of about a dime. And so what does this look like in practice? So this is seqwell, so you can see kind of the order of magnitude size. So it's about 100,000 wells on an array. And what you first do is you load these barcoded beads, and this is all by gravity. So you first load these barcoded beads, you try to achieve 100% occupancy of the beads with wells. And the beads are such, and the wells are particularly sized, so you only get a single bead per well. And the next thing you do is you add your cells. And so when we load our cells, we actually don't want to achieve complete occupancy. So we actually try to minimize the chance of having cell-cell doublets. So we only load about 10,000 cells per well. So this is what we call Poisson loading. Then we attach a semi-permeable membrane. So the semi-permeable membrane allows us to basically trap each individual cell as an independent reaction. And then we can apply our lysis buffers, so we lyse directly in the wells. We can then exchange for a hybridization buffer, which allows the beads to hybridize with the cell's RNA. We can then pool all the beads at the end, and then treat it as you would if you were doing bulk RNA prep. So this is really great because obviously this whole cycle in a BSL-3, in the absence, especially without needing a piece of equipment, takes about, let's say, 6 to 8 hours. And you can get tons and tons of information. So that's the biochemistry that underlies the method. But I would be remiss, and we'll talk about this a little bit more tomorrow, if I ignored the fact that a huge computational pipeline really underlies the ability to get really high quality data out at the end. And so the first thing you're going to do is you're going to sequence and demultiplex. And this step 2 is really, really important. Is you really want to be able to develop a model for identifying high quality cells. And so classically, these methods don't even seq well, even the best RNA sequence, single cell RNA sequencing methods don't capture every individual cell with the same sequencing depth. So you really, really need to be careful about how you decide whether a cell gets included in your analysis, because otherwise you're really just clustering noise. And then once you actually have a model for identifying high quality cells, then you can go in and you can do your cluster analysis. So as the first pass, what we wanted to ask is, perhaps there are these different subpopulations that we aren't just able to capture using classical flow-based approaches. So to really understand how maybe early events help shape the eventual immune response to TB, what we did is we took sorted CD14 matured macrophages plus minus MTB infection at a 24-hour time. And so I never really know how to transition to this next slide, but I'll just do it. So this is what the data looks like. So this is about 2,000 cells that have just been analyzed using seq-well. And what I want you to take away from this is here on the x-axis are the individual cells, and on the y-axis are different genes. And what I want you to take away from this is that there's incredible heterogeneity at the single cell level, even 24 hours after infection. And so this is really exciting, and when we actually look at defining these different subpopulations of cells, we actually don't need the full transcriptome in order to do so. For example, I'm only showing a subset of the actual genes here, but this cluster A is defined by the high expression of genes such as IL-1 receptor antagonists and TIM3. For example, in this second cluster, cluster B, it's genes that you see here, for example, promoting and SLAM-F7. And so each of these different clusters has a different set of genes that are highly expressed in a subset of cells. And to maybe show that in a more...
Brian, sir, just a clarity. So those subpopulations result from the same original population undergone similar conditions in 24 hours, just to clarify.
Yes, exactly. So these are all we know is that they were positive for bacteria, and they were all otherwise in the same culture conditions. So these are not like different wells, these are just the same well of cells that were sorted and isolated.
Different MOI.
All the same effective MOI. So we don't have a way by flow of discriminating MOI1 or MOI2. We just know that they're infected or not infected. And so just to show you that data maybe in a different way, these are violin plots. So now each column here is a different one of the clusters. And what I want you to take away from this is, for example, as I described to you, IL-1 receptor antagonist is really high in cluster A and perhaps lower in the other clusters. And for example, MALAT1 is really high in cluster G, but lower in the others. And so this is a transcriptional analysis. What we then wanted to determine was whether or not some of these transcriptional patterns could be manifest at the protein level. So for example, if we go back to cluster B here, it's characterized by expression of interferon gamma receptor. It's high SLIM-F7. We actually look by flow, we actually find that there is a functional distribution in interferon gamma receptor and SLIM-F7 expression. So we're actually, through our single cell analysis, able to corroborate, identify new markers, and then corroborate that by flow. Okay, so classically, maybe the way that we think about different macrophage subpopulations is through a polarization, M1 or M2 macrophages. So then we ask, okay, are we just recapitulating some other version of that in our analysis? So then we looked at the genes that define each of these clusters, and then we looked at known published data sets that describe M1 or M2 polarization, and actually we only found one gene. So we think that these different subpopulations are reflecting something perhaps different than the classical M1 or M2 polarization. This might be what you expect given the culture conditions that we're actually exposing ourselves to. Okay, so we've got these different clusters, but really the question is how do we mechanistically understand what is driving these different transcriptional subpopulations? So the question we wanted to ask next is how do we deconstruct this complexity? So we used a number of different approaches, and I'll describe to you two today. So the first approach that we use is we reconstructed for each individual cluster a gene correlation expression network. And so for each cluster, we have all the genes. We then create networks for each set of the genes, and then we obtain something like this. And so this is just a subnetwork in cluster B. And so the question is like, okay, how do we make sense of this? So in the mathematical space, there's actually a way to really quantify how important are particular genes to a particular network. This is called Eigenvector Centrality. So we can actually do this for every gene in the network across every cluster. And then if the clusters are actually all identical cells, the network should all look the same. And as a result, the contribution of a particular gene in a network should be the same. And so to verify that these clusters of cells that we were identifying were different, we then looked at this network centrality across different clusters. And it's very colorful, but what I really want you to take away from this is that if we look at just a subset of genes, their importance in each of these networks is quite different, which suggests, again, to us that these populations of cells are in fact very different. Okay, but what's a different way of thinking about these subpopulations of cells? So a different way of thinking about this are the genes that we're identifying that are co-clustered in these different populations consistent with one another. We might just, for example, do a simple Go analysis. And so we do that analysis, actually, in fact, we find that in each of these subpopulations, different Go terms are actually, different pathways seem to be co-segregating across these different clusters, which gave us even more confidence that perhaps we're actually recapitulating different modules and different types of macrophages that might be engaging with different processes. But this was still a little unsatisfying to us. And the reason I say this is because, okay, what's the biological mechanism that's really underlying how we might go from these different subpopulations? And so in order to do that, what we thought about is, okay, this is a transcriptional analysis, so maybe if we thought about thinking about it from the perspective of what are the transcription factors that might be underlying the activity of these different genes and the expression of these different genes. So the approach we used here is we use published ChIP-seq data and try to infer transcription factor gene relationships. So for example, now if we go back to the genes that really define cluster B and ask, do we find statistically significant enrichment of a particular transcription factor in this cluster? The answer is yes. So these are enriched for NF-kappa B targets, and it's quite enriched. It's like 10 to the minus 7. So this is really exciting for us. And I've only shown you one donor thus far, but when we actually went back and did this analysis across multiple donors, we actually find that this NF-kappa B transcriptional variability is recapitulated across multiple human donors. Really exciting to us, Marcus Covert, who's at Stanford University, has actually been studying NF-kappa B variability for quite some time using a slightly different model. So they use a tagged version of the transcription factor and watches it cycles in and out of the nucleus. And surprisingly, they find something similar with LPS stimulation. And so basically here they're just tracking cytoplasmic versus nuclear localization. And what I want you to take away from this is like in some cells there seems to be like a pulse where it doesn't return to the nucleus. In some cells, NF-kappa B remains kind of resident in the nucleus to some extent, and in some cells there's an oscillatory behavior. So while our data is only capturing a particular slice in time, we're actually able to recapitulate at least known biology in terms of the variability of the NF-kappa B signaling network. Okay, so did I just pick only one that worked? So we then decided to test this other cluster over here that I have boxed in, and in fact this was enriched for targets of the transcription factor p53. And so we were actually really puzzled by this a little bit because we, in other analyses, actually in our live versus dead sorting, came across p53 there as well. And so what we would first want to assess is whether p53 is being expressed in our cells. And surprisingly, when we did that, we found something really shocking. And that was that p53 itself was actually incredibly variably expressed in infected macrophages. So p53 is expressed in red, and so what I want you to take away from this is that p53 expression levels are actually quite different. And it's actually been shown in cancer cells that in response to DNA damage, there's actually incredible oscillation in both p53 level and activity. So this seems to be indicative of kind of a conservation of perhaps variable transcriptional networks across different subpopulations of cells. So we have this functional spectrum of MTB-infected macrophages, and I told you a little bit about NF-kappa B and p53. We actually found statistically enrichment of p63 and BRD4 in other clusters. But now the question is, how do we take these different subpopulations and place them somewhere across the spectrum? So this is kind of a poor man's way of trying to understand the contribution of these networks. What we decided to do is we took, because p53 and NF-kappa B have pharmacological inhibitors or activators that we can use to try to titrate and modulate their response, we then took these inhibitors and asked, does this really impact the viability of the bacteria? As you might have predicted, when we use our NF-kappa B inhibitor, we see a great increase in bacterial survival. But really surprisingly to us, when we use nutlin, which is an inhibitor of the interaction between p53 and its ubiquitin ligase, we see a slight increase in the ability to control bacteria. So this is really exciting, and we definitely don't think we have the answers, but we're really interested in following up how p53 responses may contribute to bacterial control. So to almost summarize, we have this model now where we think that there's different subpopulations of macrophages, some that kind of exist a priori in the restrictive state, and our kind of prevailing hypothesis right now is that perhaps it's the NF-kappa B high cells that are well poised for bacterial control, and there's some aspect of the p53 regulatory network that may or may not skew macrophages to a more permissive state. And hopefully, in the first half of the talk, I've shown you that perhaps we can recover some of the ability to control bacteria through adding GM-CSF. And in work that I haven't shown you, we actually know that for us, in our hands, when we HIV co-infect our macrophages, we actually see a loss of bacterial control. So maybe a path forward analytically is to be able to combine both cytokines, single-cell RNA sequencing, and looking at bacterial burden as a way of really understanding how these different subpopulations of macrophages contribute to bacterial control. So before I close, I just wanted to kind of ding myself on the first half of the talk, which is, I just tried to sell you on the gospel of GM-CSF, but I actually don't think that GM-CSF is the answer, or the only answer. And the reason for that is when we do this analysis, we can actually go back and look in our single-cell RNA sequencing data and ask, okay, what about GM-CSF receptor expression? The only thing I want you to take away from this is not all the cells appear to express GM-CSF receptor. So what I hope to have shown you in this talk is that single-cell RNA sequencing has a potential of unraveling so much complexity, but it also causes us to ask, what are the appropriate strategies to really think about using when we think about modulating macrophages in the setting of TB infection? And is it a one-size-fits-all approach? Or maybe do we need a polyfunctional response in order to really access these different types of macrophages in order to really resolve infection in a meaningful way? So with that, I will close. These are my friends. This is a slightly old photo of the Fortune Lab. I'm greatly indebted to my technician, Armel and Furcado, who helped with some of the ASTAing, who's now at OHSU. The Modern Lab at UCLA was really helpful with some of our original RNA sequencing work in the bulk. And the single-cell RNA sequencing would not have been possible without a strong collaboration across the Reagan, including the Shake Lab, Mark and Travis, as well as the Love Lab, especially from Todd. And I also have to acknowledge my funding sources, and I'd be glad to take any of your questions.
With that transcriptional heterogeneity that you saw through single-cell sequencing, did you do any single-cell RNA-seq in uninfected macrophages to see if the diversity is being selected for upon infection or if it's pre-existing?
Great question. So we're still mathematically, analytically trying to dissect how we think about that, but here that might give you a sense for what your question is. So we actually took the exact same cells, unexposed, uninfected, so there's no paracrine effect. So it's just like same donors, macrophages, just without ever seeing TB. And we asked about what's the pre-existing heterogeneity in that population. And so there is both pre-existing heterogeneity and heterogeneity after infection. And actually, believe it or not, there's a science paper that came out about a month and a half, independent of us, where they actually profiled CD14 positive monocytes and found heterogeneity. So I actually think that there's like multiple layers, probably starting early in the myeloid compartment, that are actually that's maybe like a feature of myeloid cells and something that we like had previously underappreciated.
Thanks so much. And I think my question is going to follow on a little bit from that one, which is it seemed like with your bacterial infections, mycobacterial infections, there wasn't that much heterogeneity amongst the mycobacterial population. You seem to have either green or red. Is that a fair assessment, or was there a lot more heterogeneity in the bacterial population than you showed on the slides?
Yeah, so there is oftentimes what I call a dual cell, where you have one bacterium that appears to be alive and one that's dead. So I don't know if that's a temporality question, is that we just caught it after it killed one and not the other. And so what we've done now is we've built a double live reporter. So basically it has two orthogonal inducible systems. So we can ask at a single cell level, okay, did this cell, like at day 4, what was going on, and then ask which of those kind of remaining viable bacteria after 4 extra days have bacteria that remain viable thereafter, to try to understand whether there's a contribution perhaps of maybe bacterial heterogeneity that might be within a given cell kind of driving some of those emergent responses.
Because just, I mean, the next thing was, you cited David Russell's early studies with Carl Roeder showing those very nice analyses of the progression of infection, where they also then looked at transcriptional profiles, right? And one of their key observations was that TB, the M. tuberculosis lineage, defined the macrophage transcriptional response. So how do you view those data now, looking back through the lens of this heterogeneity that you've seen? You've done with H37RV, would you expect a completely different profile with CDC or Beijing or any of the other?
Yeah, so my expectation is that CDC might give maybe a more muted response, just because of like how long cells that are infected with CDC seems to survive relative to H37 cells infected with H37RV. I think it'd be probably wildly different with a Beijing strain. So that's actually one of the things that we're trying to work through right now, is trying to make these experiments faster so we can, and also less expensive, to really try to understand the contribution of like different components and try to have, either some regulatable, titratable bacteria with like some titratable virulence apparatus to really understand how bacteria contribute. Because one other hypothesis is that perhaps there's subpopulations of bacteria that might be more skewing the cells to a P53 response as opposed to others, and we really can't dissect that right now with the tools we have.
Hi, very beautiful, Brian. So first, just a little follow-up to that, but in terms of residuality, we see there seem to always be a residual bacteria that seem to survive within different mutations, which is actually the same strain as opposed to, like this is the same strain, and you see a spectrum of functional spectrum of infected macrophages. So do you think actually, as you know, that actually the bacteria can actually use different mechanisms depending on what pressure they encounter from macrophages. So I've actually looked at that level, or perhaps that applying to see whether actually there's specific mechanisms at bacterial level that are actually meant for specific stressors from macrophages.
Yeah, so that's a great question. So let me tell you how we're trying to do that. Okay, so what's nice about these data and what's not so nice is that in a lot of these we actually have surface markers that we can then use, sort out the infected cells, and try to transcriptionally profile the bacteria out of those different cell populations. So that's something that we're working on now, which is technically a little difficult, to try to understand, are there bacterial responses that are associated with the interferon gamma SLIM-F7 high cells that aren't associated with the other cells. The only thing that's a little difficult is that we haven't found surface markers for every subpopulation. So we can kind of tell you about maybe this cluster here, and perhaps this cluster here. I can't tell you about the other ones yet because we don't have surface markers to isolate them in a definitive way.
How much is technical stochasticity contributing to this? So, you're doing single cells, you're not going to pick up every transcript just because of the low levels. So do you know how much that is contributing to the variability?
So, okay, so two answers. So in that analytical pipeline that we built, we actually try to account for whether or not we think a cell is sampled well enough. So so here, this wasn't even really synthetic data. So you have a lot of cells where you don't actually capture a lot of molecules. And you have a lot of cells where you capture like maybe 5,000 molecules or more. So what we did is we built an analysis which basically takes individual single cells and creates synthetic technical replicates. And we ask whether or not, at what point do we see that there's convergence between the synthetic technical replicates as a way of understanding like how many molecules for a particular cell need to be measured before we think that that cell is not too noisy and it's not zero inflated. So we actually built that pipeline, and that's actually how we determine whether or not a cell should be analyzed. So we actually built kind of a noise model. We actually toss out frankly, we toss about 80% of the cells right now in order to really feel confident that the cells that we're analyzing are not too noisy. But then, as you can see, like what I like to think about when we do these analyses is thinking about like how can we integrate, perhaps instead of looking at specific genes per se, but looking at these modules and saying that, wow, surprisingly, these modules actually point to a transcription factor uniquely in a particular cluster. Gives us even more confidence that we're actually overcoming some of the issues that are inherent to single-cell RNA-seq in the noise.
Mark, any good question?
Thanks. Have you been able to follow this chronologically to see if these sort of distinct populations converge, or whether these represent sort of different time points?
Yeah, so that's a great question. We've not yet done that yet, but kind of the next frontier is to actually really try to do a more systematic exploration. Now that we know how to get it done in the BSL-3, where we're combining our different cytokines and time points to understand what is a chronicity. Is it that they become more different, less different? Do we converge to maybe two populations as, because at an early time point I don't think that there's much bacterial killing, but maybe over time as those subpopulations of macrophages emerge based off bacterial fate, maybe we have convergence into two populations, but we're still working all that out.
So basically, you probably look at the macrophage polarization in 1 and 2 with this. And the other question is the MOI. If you play around with the MOI, do you get radical changes?
So we haven't tried to play around with MOI because our philosophy is that right now we're really interested in understanding kind of the early phases of infection where there's the bacterial burden at a single cell level is pretty low. But we are really interested in understanding kind of how we can use this model to understand, higher levels of bacteria per cell and more complexity, but we haven't looked at it. We have definitely not looked at the influence of MOI, but my expectation is that what I see by ELISA and looking at other MOI effects, I think there's definitely going to be a reorganization transcriptionally because of the paracrine response that would emerge in addition to just having more bacteria per cell.
There's another question. You do this pathway analysis via activation score. Very, very interesting. But I'm sure you found more than GM-CSF.
Oh yeah, there's like tons of pathways, and this is actually one of the things that really got us excited about the single cell approach is perhaps it's not that every cell is using the same exact pathways. Maybe there's an evolutionary reason for diversity, right? If you think about it, if you only have one strategy for killing bacteria, and the bacteria learn about that strategy, you're kind of screwed. And so what we like to think about is maybe these different, maybe there are pathways that actually go together, and we just see like high activation of multiple pathways, but they're all happening in the same cell. Or perhaps using the single cell RNA-seq, we can try to deconvolve whether or not maybe some cells are turning on a certain pathway in order to drive control, and some cells are turning on a different pathway. So yeah, so the long and the short answer is there's a lot of pathways. We're still trying to dissect some of them. I would say that the caveat of this analysis is that it's kind of chicken and egg. When we look at 4 days to ask what are the pathways that are different, because you could imagine, and we found this, that some of the pathways are just, I killed the bacteria, or I have active bacteria inside of me, and they might actually be causally associated with bacterial control. So they might just be reflective of being infected or not.
I'm going to go there.
Hi, thank you. This is a very interesting talk. So I've got 2 questions. So one is, you talk about the story, I mean, in your initial story on GM-CSF, you showed that there's also an activated inflammasome. So I guess, were you actually going through each individual pathway that GM-CSF activates, or was it more of a global RNA-seq approach that you did?
So this was a totally unbiased approach. So actually, if we go back to our analysis and look at either macrophages stimulated GM-CSF, or we look at other pathways to maybe your point, we actually find IL-1 beta-driven transcriptional signaling high in the cells that kill TB as well. So at least we're seeing this kind of co-segregation of GM-CSF and IL-1 beta signaling in the same population of cells. But the way that we did our small molecule screen is we basically said, what are the different pathways that we know contribute to bacterial control or are downstream of the GM-CSF receptor? And we tried every drug we could, and whichever ones killed macrophages in the 4 days of the experiment, we kind of had to toss out.
Because I mean, obviously, IL-1 beta also has its own independent way of killing bacteria.
Yep.
From Archer as well.
Yeah.
So the second question was, I mean, generally, what do you think is the actual role of GM-CSF in terms of disease progression in MTB?
So that's a great question, and it's not clear to me, like, explicitly what GM-CSF is doing to, there's like multiple different effects. So I think that GM-CSF globally at the level of the host might be doing something very different than what it's doing to macrophages. And so at the macrophage level in the setting of infection, I would argue that it's probably driving the cells to a particular more inflammatory state, and that's what many people have already shown. But in the setting of like in a lesion, we don't know what the GM-CSF levels are, so it's like hard to like explicitly say what it's doing. And we think in the mouse model, it obviously shows one phenotype, but I think that's a gross phenotype, and knowing exactly how GM-CSF is contributing in the setting of infection. I mean, people have shown that GM-CSF levels at the serum level are lower, but I'm not sure like how relevant that is. So my assumption is that GM-CSF is pushing the cells to a more inflammatory state, but it could be being antagonized by a number of different factors at the same time.
This variability, we also explained by the localization of MTB within the cells. For example, some MTB might be found in the autophagy pathway, the other one lysosome, the other one would have escaped. And in other words, could you also look at these different localization?
So combining that, so I think, my short answer is yes, but not with single-cell RNA sequencing as it's done right now. So now that we have markers that we think that we like, we can then go back and either do FISH or IF for those markers and trying to capture the localization information. because right now the single-cell RNA sequencing is pretty destructive. So as soon as you've lysed the cells, you kind of lose all that spatial information. But what we're trying to do now is combine the ability to spatially resolve infection with kind of spatially resolved transcriptomics to understand some of those dynamics as well.
Okay, last question from Jackson.
Yeah, so in terms of your networks of variability, you showed, so your core terms showed interferon inducible signaling of some sort of signature. Did you try and enrich that with chip the same way you did with P53 and
Yeah, so in terms of the kind of the Go terms, we kind of left them they were kind of an independent analysis of the transcription factors. In terms of transcription factors that are kind of classically associated with interferon signaling, we didn't find anything that really achieves statistical significance. But that doesn't mean it's not there. It could just be, the integration of the signal wasn't enough to get us over those borders, but we haven't fully found kind of an interferon transcription factor that's connected with those clusters quite yet.
Sure. And is there some form of validation like I have in some of your candidates and so on?
Yeah, so for a lot of these we've done flow to show that it's not, so what I will say is that for different single-cell RNA sequencing analyses, sometimes it's a spectrum and sometimes it's obvious subpopulations. I would say that when I've taken different populations of T cells, the analytics, just because you're dealing with kind of zero inflated data, it's not as night and day as if you just did classical transcription factor staining. So, so my short answer is a lot of these transcription factors we've at least looked for P53. For the NF-kappa B story, we actually have the NF-kappa B reporter that I told you from Marcus Kover's lab. So we've done live cell imaging to look at NF-kappa B dynamics, and that's consistent. So we feel pretty confident about those. We haven't tested the others yet.
Okay, please join me in thanking Bryan. And if you want to talk to Bryan during the week, come and let me know.
Yeah,